Hey everyone, how's it going? This is the last PowerPoint for Module 1. This is Module 1.32, and we're still talking about the chemical cycle of organization, specifically macromolecules in this setting. What I want you to take away from this is to understand the properties, uses, and examples of carbohydrates, lipids, proteins, and nucleic acids in the human body. And we're going to cover this briefly, as much as you need to know for Bio 168. A lot of these will come more into play as you get into 169, and especially if you move on to higher levels of science like biochemistry and things like that. So as I just discussed, there are 4 categories of organic compounds that are important to the human body. Those include carbohydrates, lipids, proteins, and nucleic acids. Organic compounds are typically polymers that are constructed of monomers, so each of the categories that I just listed has a subunit classification. Okay, and these always contain some sort of carbon, hydrogen, and generally an oxygen, as well as several different other subgroups. Many are long chains of carbons that are linked with covalent bonds. A lot of organic molecules are. Several of them are soluble in water, which helps them interact with each other inside the human body, and we'll talk about one in particular that's not. Functional groups are a large component. These are attached groupings of atoms that occur commonly in these organic molecules and can influence the properties of the overall molecule itself. And many allow cells to transfer and capture energy as high energy compounds. We're going to talk about 2 in particular that are going to be highlighted on your quiz that are directly involved in energy storage and consumption. First up is the carbohydrates, and these are organic molecules, as we already discussed, containing hydrogen, carbon, and oxygen in a ratio that's generally 1 to 2 to 1. Example of carbohydrates are going to include sugars and starches, sugars being usually a simple carbohydrate, or starches being a more complex, and these are roughly 1.5% of the total body weight and serve mostly as important energy sources. They allow for quick energy usage in the body. You have a few different subcategories of carbohydrates. These include the monosaccharides, which are your simple sugars, things like glucose and fructose. You have your disaccharides, which are a combination of your simple sugars and monosaccharides, such as sucrose and lactose. And you have your polysaccharides, which are your larger chain, more complex organic carbohydrates like glycogen and things like that. So your monosaccharides, these are usually a single ring structure of a carbohydrate. Things like glucose and fructose. These are what your body breaks down those complex carbohydrates to form these molecules. You can see several examples here: glucose, fructose, galactose. You see most of these have roughly the same structure, just a few minute details in comparison. Some of these are 6 carbon sugars. You can see the carbons. I'm not sure how many of you have had an overview of some of these ring structures that you would see, such as in organic chemistry or things like that. But if you can count the number of points here, each one of these points represents a carbon. Don't worry about the bolded lines. That has to do with stereochemistry. We don't need to worry about that. But each one of these points here represents a carbon. So if we count the number of points here, we can tell that this is a 6-ring carbon, or a 6 carbon sugar. So 1, 2, 3, 4, 5, 6. And then an example of a 5 carbon sugar would be fructose. So 1, 2, 3, 4, 5. So this is a good time to talk about isomers. Isomers are molecules with the same molecular formula, but they have different structures. An example of these would be fructose and glucose. So if you look at fructose and glucose, both have the same molecular formula of C6H12O6. However, you can tell by the structures that the ring structure is a little different. Glucose being a 6-ring sugar and fructose being a 5-ring sugar. Alright, as we move up the level of classification for carbohydrates, we move into the disaccharides, which are usually combinations of your monosaccharides. So an example of this would be sucrose. These are formed usually by a dehydration reaction that creates a disaccharide, which means that water is removed from the situation, meaning that these are very soluble in water. Hydrolysis usually breaks them down for even further. This is an important physiological process, and breaks them down into those monosaccharides. And if you break down the word hydrolysis, if you don't know what it means, if you look at those prefixes and suffixes list that I gave you on Sakai, you'll see hydro meaning water and lysis meaning to break apart. So hydrolysis would be breaking apart using water. We can move on down the list or up the level of classification here and get to polysaccharides. So if saccharide meaning sugar and poly meaning multiple, and these are complex carbohydrates that form from multiple disaccharides and or monosaccharides. So these are long chain polysaccharides. Think, for example, starches, which are formed by plants from glucose, and glycogen, which are usually in the form of animal starches. There are usually three important polysaccharides when we talk about physiologies. You have starches, glycogen, and cellulose. All right, moving on to lipids. Lipids generally contain carbon, hydrogen, and oxygen. That seems to be a theme here. However, their carbon-hydrogen ratio is nearly 1 to 2, so much less oxygen compared to carbohydrates, but with a simple number of carbon atoms, and usually contains some other quantity of some other type of element, whether it be phosphorus, nitrogen, sulfur. Example of these include your fats, your oils, your waxes, anything that doesn't really mix or play nice with water, and that's highlighted there, so most are insoluble. And these play a very important role in a lot of physiological systems to prevent certain molecules from crossing over barriers, so they are very important in barriers such as a phospholipid bilayer, hence the name phospholipid. And we'll get into that further as we get into the structure of some organelles and things like that. But they also serve other purposes, such as to provide energy for cell structure and insulation. We'll talk about cholesterol and the importance of that as we get further as well. They usually contain two parts, usually the head, which is a glycerol molecule, and the tails, which can be considered fatty acids. The monomer of this is the fatty acid. So this just highlights a little bit more of the lipids here. So generally these are important as energy reserves, where your carbohydrates are going to give you that quick burst of energy that you need, that sugar rush, if you will. Your lipids are going to provide almost twice as much energy as carbohydrates, but they're stored energy, so it uses more to convert them into usable energy, and they account for normally 12 to 24% of the total body weight. And they have several different functions other than what we mentioned. Again, the chemical messengers. They are components of cell structures, think the phospholipid bilayer, and I give you some more examples here as well. This is just some pictures of this, and we'll talk about saturated versus unsaturated fatty acids here in just a little bit. So saturated versus unsaturated fatty acid. These serve different purposes in the body. I'm sure you've heard of good fats and bad fats and what those really mean. And there's a lot of complex things that you can go into here that I've listed over here to the left side of this slide. So that saturated fatty acids, each carbon in the tail has 4 covalent bonds, and unsaturated fatty acids include 1 double bond equal monosaturated, and so on and so forth as you move forward into polysaturated and multi-polyunsaturated and a bunch of different things like that. But basically what you need to look for when you're talking about saturated versus unsaturated, if we go back to this diagram here, you can see the inclusion of the double bond there. When you start to see double bond and kinks in the tail, as it's called, you'll start to get into your unsaturated fatty acids. And basically what happens when that happens is that the double bond changes the shape of the tail, which changes the shape or changes the way that the body metabolizes the fatty acid, or how that fatty acid interacts with different molecules in the body. Start talking about different forms of lipids here. So your glycerides are more commonly known as your acylglycerols, or your esters formed from glycerols and fatty acids. Let me remind you guys that a lot of this stuff is above and beyond what I'm going to require you to know. So this just serves as a reference for you. So you can go back and look at these things when you start to get into different levels, or if you want to learn more about these things and to see how they play an important role physiologically. But I may not test exactly on some of the minute details that are found in these slides. Like glycerol has a 3-hydroxyl functional group. I'm assuming, or going under the assumption, with this being a 100 level course, that most of you don't know what a 3-hydroxyl functional group is. that's okay. I'm not going to test you on that sort of thing. But I would like you to know that a glyceride is a form of a lipid. Because you're going to hear things as you move forward like monoglyceride, diacylglyceride, and triglycerides. I'm sure you know that vegetable oils and animal fats contain mostly triglycerides, which are going to have these 3 fatty acid tails with a big old kink in the bottom here. As we talked about, this is your saturated, and then unsaturated here as the inclusion of the double bond. Phospholipids are largely functional group of the phospholipids, and they're a class of lipids that are a major component of all the cell membranes. We'll talk about that more when we get into organelles, but they can form bilayers, they can form monolayers because of a particular quality that they possess. These phospholipids are typically amphipathic, meaning that they have some hydrophobic qualities and some hydrophilic qualities. So the structure of the phospholipid molecule generally consists of a hydrophobic fatty acid tail and a hydrophilic head that contains the phosphate group. So when the two components are joined together, they're usually joined together by a glycerol molecule. So if we break down what those words, those hydrophobic meaning that it's water fearing, and hydrophilic meaning water loving. So the head wants to interact with water, the tail wants to avoid water at all costs. So what will happen is when you introduce phospholipids into an aqueous or a water environment, they'll start to form together so that they can protect those hydrophobic fatty acid tails. And what will happen is they'll form in certain conditions, or at least when this was first observed, what are called micelles, which were just a single phospholipid layer where the tails point inwards to protect the tails from the aqueous environment, and the heads, which are hydrophilic, are pointing outwards. Glycolipids are also lipids with a carbohydrate attached to the glycosidic covalent bond. So you'll find a lot of these in the phospholipid bilayer as well, and their role is to generally maintain the stability of the cell membrane and facilitate that cellular recognition, which we'll talk about a little bit later, which is another function of peripheral proteins as we get into that. Like I said, they're found on the surface of most eukaryotic cell membranes, where they extend from the phospholipid bilayer into the intracellular membrane. I'll highlight that more as we get to the phospholipid bilayer in the organelle section. Lipids also have another structural classification known as steroids, and these are usually large molecules with 4 carbon rings and differ in attachment and functional groups. Functional groups meaning the things that hang off of them. So basically what that means in a nutshell is that they're all going to have that same basic outline. So you're usually going to see this outline, which is the cholesterol molecule that serves as the basis of most steroids. So you're generally going to see, when you talk about steroids, the same sort of looking structure every time, but the stuff on the ends, so your functional groups, that's what those are called, are going to be a little different. Notice that cholesterol has this structure. You can see the same general structure in estrogen and the same general structure in testosterone, which are both cholesterol derivatives as steroids. And you'll get more into what those two hormones are when you get into 169. But you can see a lot of the stuff changes on the outside of them. Estrogen and testosterone lack this big functional group chain that cholesterol has here. You can see the inclusion of some double bonds here that are missing here. You can see the same thing here. Eicosanoids will also be important as you move into 169, and a lot of these are derived from arachidonic acid. There's an important metabolic pathway that you'll get into when you get into 169 as well that involves leukotrienes and prostaglandins, but we're not going to talk about that in detail here in 168. Proteins are the most abundant molecule in the human body and are very, very important and account for about 20% of the total body weight. Okay. The monomer of the protein is the amino acid. There are about 20 amino acids in the body, and several of them, I believe the number 7, I could be wrong on that, but are the fundamental amino acids. If you move into biochemistry and things like that, you'll have to memorize all those, so be prepared for that. Most proteins, like the other macromolecules, are going to have the same general structure components, but proteins differ in that they are usually a central carbon that are attached to 4 different groups. So these will be your functional groups here. Your hydrogen atom, which you can see here, your amino group, which is on the side here, your carboxyl group, And for those of you who haven't taken organic chemistry or chemistry 2 or anything like that, the carboxyl group is just going to be your double bonded oxygen there with an OH group on the other side of that carbon. And then an R group. And then whenever you see an R group in chemistry, that's just a variable side chain. Think of the X variable when you talk about X in algebra. Your R group is the same thing in chemistry. But in a protein, the molecule has both positive and negative charges, so those equal out into a net charge of 0. And it's that different R group, this different R functional group here, that variable side chain, that's going to give each amino acid its special chemical properties. You have dipeptides, which are usually 2 of these put together. Think di, 2. Polypeptides, which are going to be multiple chains. These are peptides containing over 100 amino acid chains long. Proteins can have up to 4 levels of structural complexity, which we'll get into here. You can have your primary structure, which is usually just a single strain of a single polypeptide strain. You can get into a secondary strain, which results from some different bonding techniques. So you start to form hydrogen bondings between some of the different functional groups in the proteins, and it starts to cause some folding and some twisting in the structure of the protein. Like I said, so you'll have some hydrogen bonding, there'll be some van der Waals forces in there, some other different types of forces. And these can create usually 2 distinct features when you talk about secondary structures, which are your alpha helices, which is just a spiral, and then you'll form what's called a beta pleated sheet or a flat pleated sheet, and you can see that here. Your tertiary structures, where you start to get some of the more complex functional usage out of your proteins, and these start to form such a 3D shape, and these are usually interactions from the protein and surrounding water molecules with some of those hydrophobic properties. And you also get some contribution again from those R groups that you see on the side there. So a lot of times you'll see different sorts of your secondary structures, your singular structures here that start to form that 3D structure. As highlighted in this diagram here, you can see an alpha helice, and then you can see a lot of polypeptide chains here, that singular structure. Your quaternary structure is going to be a lot of different interactions from a lot of different tertiary structures that form what's called a protein complex. And these are going to be your most functional, and these are highly specialized molecules and compounds in the body. Think hemoglobin, collagen, things like that. Denaturation can occur in proteins, and this is usually a change in the protein's tertiary quaternary structure. And that protein shape is going to change its function and work to deteriorate the protein. These occur under extreme conditions. Several conditions can affect this, such as pH, temperature, tonicity, which is usually the salt or solute content, and several different other things. This can be fatal due to the denaturation of structural proteins and enzymes, because when that happens, you lose a lot of the functionality of that protein. And a lot of this can cause irreparable damage, because although they can reform when that high stress environment is removed, sometimes they don't reform in the correct way. This is especially important when we start talking about enzymes, which are specialized proteins that help to facilitate a lot of the metabolic reactions inside of the body. So when you talk about enzymes, you have a lot of different types of vocabulary, such as the substrates, which are the reactants in an enzymatic reaction. You have the active site, which is where that substrate is going to attach to. And then usually have your product, which is going to be whatever comes out of the reaction from the enzyme and the substrate. You can see this nice little diagram I included down here, which shows a substrate attacking to the active site, the enzyme substrate complex formation here, the beginnings of the formation of the product here, and then the release of the product from the active site of the enzyme. One thing that I don't cover here that I would like you to know is that enzymes are very highly specialized. So usually they only interact with one type of substrate, which helps control what that enzyme does and where it interacts in the body. Meaning they're usually seen with a specific shape. Notice how this substrate here has a specific shape that directly corresponds to the active site of this enzyme. You can see the steps listed in an enzymatic reaction here. All right, and our fourth and final macromolecule that we're going to talk about in 168 are the nucleic acids, which are generally large organic molecules that are composed of carbon, hydrogen, and oxygen, again to be that theme. But these also include a little bit of nitrogen and phosphorus. There are three general categories of your nucleic acid. You're going to find your deoxyribose nucleic acid, which is your DNA, your ribonucleic acid, which is your RNA, and your adenosine triphosphate, which is your ATP. Most of you probably remember what all three of these are from biology 1 or 2, or if you took chemistry 1 or 2. But the primary function of these is the storage and transfer of information. In particular, for DNA and RNA, your genetic information, and an ATP is usually the energy molecule of the body. A lot of these consist of 1 to 2 long chains that are formed from dehydration synthesis of the subunits. Remember that dehydration is the removal of water from the system. And usually we call these nucleotides. You see the structure of the nucleotide off to the right here, which is generally going to be formed from a nitrogenous base, which you probably learned about in Bio 1. Those generally include your adenine, your thymine, your guanine, your cytosine, that A to T pairing, that G to C pairing that you remember from your study of DNA, a sugar molecule of some sort, which you can find in your deoxyribo here and your ribonucleics. So those are going to talk about that here. If we were being specific, we could count the number of carbons like we just talked about a little while ago, and notice that this is a 5-ring structure. Ribonucleic acid, or ribose, is a 5-carbon sugar. So it has a 5-ring structure here. So I know that this is a ribonucleic acid based off of the 5-ring sugar here. If it had 6 rings, it would be deoxyribose, which is a 6-ring sugar, which would have 6 points on the end here. And then the final subunit of a nucleic acid is going to be a phosphate group down at the bottom here. Nucleotides serve as the building blocks of a lot of those larger nucleic acids, which help to include several things like DNA and RNA. You can see listed here those CT and U pairings. U is going to be the one that's included in RNA that I was talking about earlier, and then your adenine and guanine here, which are your purines. There are 2 subsites of those nitrogenous bases that you talk about when you talk about DNA and RNA. You have your pyrimidines, which is your C, T, and your U, and then your purines, which is your A and your G. Notice how the pyrimidines have the same general structure, and your purines have the same general structure here. Again, you'll get into way more details if you go into this stuff in chemistry 2 or in organic chemistry / biochemistry. We already talked about DNA and RNA being nucleic acids, and generally the phosphate and sugar adjacent nucleotides bound together by dehydration synthesis. I already talked about that dehydration as well. This usually produces a chain backbone of a sugar-phosphate-sugar sequence with the nitrogenous base that projects inward to one side of the molecule. So you have that sugar-phosphate backbone, which I'm sure you remember from biology, and that interlocking nitrogenous base sequence that you remember we talked about those base pairings. You talked about that in biology, but it just highlighted that A to T pairing, G to C pairing in DNA, that A to U pairing, G to C pairing in RNA. This helps to give DNA and RNA that stereotypic structure that we're used to seeing when we talk about these molecules. For DNA, it's that double helical structure. And for RNA, it's usually a single helice that usually formulates down onto itself. But RNA can be single-stranded or double-stranded. Typically, when we talk about RNA at this level, we're talking about a single-strand RNA. RNA has a few different subgroups we can talk about here that are going to be important when you get into the cell replication cycle. And these are going to be your messenger RNA or your mRNA, your transfer RNA or your tRNA, and then your ribosomal RNA or your rRNA. I'm sure you all know this already, but ATP is considered the energy currency of cells and is highly important, especially when we talk about reactions that go against a gradient. ATP can exist in a few different forms as well. You can have ATP, which is adenosine triphosphate. You can have AMP, which is adenosine monophosphate. adenosine diphosphate, which is ADP, and a lot of those you're going to see within the cell cycle itself, or at least within the Krebs cycle / the electron transport chain. No need to go too in-depth into that. I'm never going to require you to pick out what each one of these molecules is and what's actually happening here,